I'm the editor-in-chief of MPMI Journal. And this is What's New in MPMI, which is our virtual seminar series where we highlight different papers from MPMI. We make them all open access so that you can come and follow up if you want to go a little deeper. And we pick topics, I select topics that I'm interested in. And this particular one, I'm delighted to have Antonio Molina here today talking about one of his. Oh, and I should point out that Algae Extracts is my co-host today. She is a senior editor on the MPMI editorial board, and Algae is one of the guest editors of our new focus issue coming up, looking at how do plants interact simultaneously with beneficial microbes as well as with pathogens. And she's co-editing that with me, with Dong Wang, and with Kay Jones. So I really hope that people will be looking at that and submitting articles or reviews, short communications to our focus issue. So I hope that will come. We will need to have submissions in by the end of October. Okay, and yes, so on to our seminar. Antonio Molina, I'm delighted to have him here. When I picked his paper, the paper that Laura Bacete and colleagues wrote, it's interesting because it's taking a look at the cell wall as a real component in the signaling between plants and microbes. I mean, it's obvious, it's the outer part, it's the point of first interaction. And but we've known for years that it was important, but what are the components? What are the pieces? How does that work? And this story ties those cell wall fragments into defense and cytokinin signaling. So that was interesting. And don't just take my word for it. It was picked up by 7 different news sites, which was exciting. And I saw one when I opened up the email from Biotechniques and saw it highlighted there as one of their interesting science articles that came out recently. And that was surprising because they don't do that for plant papers as often. So that was exciting to see. Also been blogged by one on a Wikipedia page. So it's attracted a lot of excitement. And we're going to hear today about that paper, but also where is that going? And this is important because one of the top 10, we tried to investigate what are the top 10 unanswered questions in MPMI. And one of them is, how do you translate results from basic science research into agriculture? How do we make that change? How do we make that connection? And what's beautiful about this is you're going to see the whole arc here, the story arc from the basic science all the way to how could it be used in the field. So I'm going to stop there and I'm going to turn it over to Antonio, who's going to present the paper.
Okay, so good morning, good afternoon, and good night, because I saw there are people from the Philippines. It's really a pleasure to be the invited speaker today for this MPMI virtual seminar, and I will start to share my presentation. And as I said, it's a pleasure to be here and to have the opportunity, as Jane mentioned, just to describe how we start to characterize Arabidopsis resistant to different pathogens and how we reach the point that the cell wall is a key element of plant immune response. So actually we are publishing soon in Interaction in MPMI, this special issue of MPMI Interaction, the whole history of how we reach this point, and the reason my group is moving from plant resistant to necrotrophic fungi to a group that is mainly focused on the contribution of plant cell wall to plant immunity. So the paper that we will discuss today is our recent paper that was published in IS-MPMI, Arabidopsis Response Regulator 6, ARR6, modulates plant cell wall composition and disease resistance. And the first author of this paper is Laura Bassete, that I guess is she's online. This work has been done together with some of our collaborators, the group of Vincent Boulant, the group of Deborah Goffner, and also with Dominique Tremousseyer. And in the research activity in our center is in the Center for Plant Biotechnology and Genomics in Madrid. That is one of the center of excellence of the Spanish Research Agency on the Spanish system. And I will also invite you to visit our webpage and to see the different activities and research project that we are developing here. I would like also to thank Ashley for this fantastic press release that Jane already mentioned, that has a lot of media impact, and I would like to focus on this particular title: Cell Wall Research presents possibility of a simple and sustainable method to protect crops, because at the end of my presentation I will show some examples of the potential of the use of using the cell wall and the knowledge that we are acquiring in the study of the contribution of cell wall to plant immunity in a potential application to develop a method for a more sustainable agriculture. And just before to start with the paper, just to mention that, as I said, this research, this article was mainly done in my group, that is the Plant Immunity and Resistant to Necrotrophic Fungi group, the CBGP. And the main contributor of this paper in my lab have been Laura Bassete. This was part of her PhD. Hugo Melida, a very talented postdoctoral in the group. Our technician Gemma Lopez, and Eva Mieres. And you can see here the rest of the members of the group. And as I mentioned, we have collaborators, and I would like to also acknowledge the financial support from the Spanish Research Agency and the other agencies from the other collaborators. Well, as you know, one of the key elements of plant immunity is the recognition of microbes and pathogen. And to do this, the plants have developed two layers of recognition. One of these layers, as you know already, consists of a set of pattern recognition receptor, some protein receptor that are able to perceive microbe-associated molecular patterns, or pathogen-associated molecular pattern is the two names that we use for this, derived from the pathogen or the microbes. And this recognition leads to the formation of a complex with a co-receptor, a co-PR, and this activates a plant pattern trigger immunity. And this is a key element of the immune response in plant. And this activates different response, like the regulation of genes and all the immune response. In addition to this immune response that is activated by non-self molecules that are derived from microbes or pathogen, there is also another set of receptors of PRRs that are able to perceive self compounds that are derived from the plants. And among these self compounds are peptides, but also carbohydrate or glycans that are derived from the cell wall. The majority of these compounds are referred as DAMs, or damaged associated molecular patterns, and are released upon infection of plant by different pathogens, of interaction with different microorganisms, and as I will try to show you today, also by perturbation of the cell wall that can be caused, as I mentioned, by pathogen or also by other stress. This additional layer of defense or of immunity we call DAM trigger immunity, or DTI, and it's not clear at the moment if these two branch of the immune response are identical, or they show a lot of difference at the molecular level. In addition, as you know, there is another layer of recognition that is based on the recognition of effectors, previously known as a virulent protein. And these effectors are recognized by intracellular receptor, the R gene, and this recognition activate the effector trigger immunity. And some of these effectors, also is really well known, is inhibiting the activity of PTI. Okay. So these PRR plants have hundreds of these PRRs, and they are different structured ectodomains, extracellular domains on this PRR that recognize different types of DAMs or MAMs. One of the major groups is composed by the PRR with LRR domains, and this structure mainly recognize peptide, but not only peptides. And there are other groups that have been described to potentially bind other compounds. In some cases, this has been confirmed. But some of these ectodomains has been shown to recognize some glycan, like for example, those from the Lys-M family that recognize of bind chitin, also beta-1,3 glucans, peptidoglycans, or those from the EG-like family, like WOCTO, that is able to perceive OGs. This recognition activates a very complex immune response that I will not go into much detail. You can see this in the review that we published in 2018 in the Plant Journal, that the first author is Laura Bacete too. And open this recognition, there is an activation, for example, in the case of LR, that imply the back and the SER co-receptor. In the case of some PRR without kinase domain, also this involves the formation of complex with SOBR1, and this activates different early immune response, like the increasing calcium cytoplasmic calcium, also the activation of ROS production, the phosphorylation of MAP kinase, and the upregulation of some immune genes. I will not go into detail of this. This was just to give an example of the type of response that we are measuring in the characterization of the immune response in the mutant that I will describe in the following slides. So this recognition of the pathogen also activates the synthesis of some hormones, and it's well known that jasmonic acid and ethylene are required for the resistance to necrotroph, but also salicylic acid is required for this resistance to biotroph. In addition to these classical hormones, other hormones has been shown to be involved in the activation of the immune response, like abscisic acid, gibberellic acid, brasinosteroid, auxin, and the group of hormones that I will focus today, that are cytokinins. And the contribution of this hormone to the resistance to different pathogens has been really well characterized, but in some cases there are a lot of doubts regarding the function of these hormones. And I will try to explain what is our view now of the contribution of cytokinins, and particularly the Arabidopsis response regulator that is part of the cytokinins pathway in the regulation of the immune response to necrotroph and vascular pathogen. And one element that probably is the first defensive element that I guess has been understudied, probably because it's very complex to study, is the cell wall. And the cell wall is clearly a physical barrier to pathogen infection. And as you can see here in this very fancy cartoon, also from Laura, it's a very complex structure. All the plant cells have a primary cell wall, and those cells that have completed their expansion, or of course also that have a specific function, like in the vascular system, have a secondary wall. And this primary and secondary wall, as is shown here, is very complex. It's mainly composed of cellulose and pectins, and also Emi Cellulose with different components that I will not go into much detail. And in the secondary cell wall, there is also an accumulation of lignin. And the contribution of the cell wall clearly in the last year has been shown that it's not just as a physical barrier, but the cell wall is also has a function as a cell wall monitoring system, and there is a cell wall integrity monitoring system, and where there is an alteration of the cell wall integrity, this is sensed by the plant, and this is this result in the activation of some immune response. And also the cell wall is a source of secondary metabolites and protein, and that is the subject of this seminar, a source of signaling molecules, and in particular from DAMs, and in this case, glycans. So we know very well that the alteration of the cell wall, as this is shown in this cartoon, for example, of the cellulose content, pectin, Emi Cellulose of callose synthesis, affect the plant resistant to pathogens. So this is clearly an example of the contribution of the cell wall to the immune response. And modification of this in the composition of the level of these different components of the cell wall result in the activation of some immune response that lead to enhanced resistance and in some cases enhanced susceptibility to some pathogen. But it's not clear what are the reason that this alteration in the cell wall result in this activation of the immune pathway. Because in some case, we could think that this alteration in the cell wall will result in the change in the structure of the cell wall, and this will affect the capacity of some pathogen to degrade the cell wall. But our initial hypothesis after the characterization of several cell wall mutants was that the modification of the cell wall may result in the release of some DAMs that will activate the immune response. And I guess that the last data that we got, including the data of the paper published in MPMI, just confirmed this initial hypothesis that we had. So I will skip this. So based on this initial idea, so what we decided to do a long time ago, almost 10 years ago, it was to follow a very crazy approach, and we select a lot of Arabidopsis cell wall mutants or putative cell wall mutants. Some of these cell wall mutants were very well characterized in Arabidopsis, but in other case, we just select genes, ortholog of genes of Cynia elegans that were expressed during the phylogenesis that we know is a process that involves the formation of secondary cell wall. So we select a lot of mutants, in some cases, as I said, very well characterized cell wall mutant, in other case, putative mutants, and we just took this big collection of mutant and we checked the resistance of this mutant to 4 pathogens that represent probably the main group of pathogen in agriculture. So we use Plastisphera cucumerina, the necrotrophic fungus that we work with. Actually, we just published also in MPMI the genome sequence of this guy and 2 additional strains, and also the transcriptomic analysis of this, the interaction between Plastisphera and Arabidopsis. You want to look at the paper. We use also the Rastonia solanacearum, that is a vascular bacterium. Now it's called Rastonia pseudo-solanacearum. The biotrophic Omyces Yalloperonospora Arabidopsidis, and the biotrophic fungus Erysife cruciferarum. And we check the resistance of a bunch of mutants, and these data are not all then here. And we found this astonishing result. And this result is really surprising because we found that 70% of the mutants that we tested show alteration to at least one of these pathogens. And this was very surprising for us, that we have performed several screening of mutant using EMS population, and you are lucky you identify one mutant among 10,000 that show alteration in the C-resistant phenotype. And here, 70% of the mutant that we checked, we saw difference in the level of resistance. In this scheme, the orange color means more resistant than wild type, and the blue color more susceptible than wild type, that is the white color. And we focus in a couple of mutant alleles that we identified that are mutant in the Arabidopsis response regulator 6. That is a group of protein that has been involved in the cytokinin pathway. And since the role of cytokinins in immune response was not well characterized, we decided to characterize this group of mutants. By the way, we have recently just published in the archive an article that is under review in another journal with the description of this approach that is explained in this slide. So interestingly, the Arabidopsis response regulator, when we look at the data of expression in the database, we were a little surprised because the expression of this gene, as you can see here in this scheme, that is actually the figure supplementary 1 of the paper, is that this gene is clearly, the expression is downregulated after infection with compatible and incompatible pathogen, and interestingly also after treatment with mumps and also after in response to several abiotic stress. So what is this Arabidopsis response regulator? What is the function? Well, these are components of the cytokinin pathway that is summarized here. So in this cytokinin pathway, the cytokinins are perceived by the cytokinin receptor that are HK2, HK3, and KRI1. And this recognition by this cytokinin receptor that are histidine kinase just phosphorylate this Arabidopsis histidine phosphotransfer protein. And this phosphotransfer protein just phosphorylate the Arabidopsis response regulator in the nucleus, and this activate the expression of some additional Arabidopsis response regulator type A, and this also this activation result in the activation of the cytokinin response. As you can see here, there is also a feedback negative loop of the type A ARRs on the type B. So this Arabidopsis response regulator is a very complex family. So actually there are more than 20 members, and as you can see here, the ARCs that we are characterizing belongs to type A, and this group of, together with number 5, 15, and 16, has a particular feature. This AR6 has been previously shown by the group of Chris Argueso. You can see this in her web page, that probably the cytokinin, the hypothesis of the group of Chris was that the cytokinins are activating salicylic acid pathway, and that type ARRs are a negative regulator of the Arabidopsis response to the Hyaloparanospora parasitica, this biotrophic acid that I mentioned. And this is mainly the result of a generation of overexpressor line of several type A Arabidopsis response regulator, and this overexpression line show enhanced susceptibility to HPA. Individual mutant in each of these type A, they don't see any phenotype because there is a overlapping function, and only in a sextuple mutant they realize that these plants show enhanced resistance to slightly enhanced resistance to Hyaloparanospora parasitica. So based on this previous data, what we did first was to characterize two mutant allele, the allele 6.3 and 6.2. 6.3, as you can see here in this QRT expression analysis, is a knockout mutant, whereas the allele 6.2 is a knockdown mutant. And we generate transgenic plant overexpressing the AR6 either with a different protein tag like GFP-HA, and we overexpress this ARR6 under the control of 35S promoter in wild-type plant that you can see here, and in the mutant background. In this, from the bunch of transgenic line that Laura analyzed, we select some lines that you can see here that are the line that we have used in this paper. So we check the mutant and the transgenic line with the different pathogen, and this you can see here the result. That is the figure 1 of the paper. So first, in the case of necrotrophic fungus, that we determine the growth of the fungus just by analyzing visually the seed rating, but also determining the fungal biomass by determining the level of beta-tubulin expression from the fungus. You can see here that the wild-type plant, that is Columbia, has this level of fungal biomass. The knockdown allele of Arabidopsis response regulator 6 show enhanced resistance, whereas the overexpression line show enhanced susceptibility. Interestingly, the the overexpression of RR6 just to restore the wild type level, as you can see here in the QRT-PCR analysis, just restore the enhanced resistance of the mutant to a wild type level. These two lines that you see here are control lines that actually are mutant affected in the cell wall that we have characterized previously in our lab. This is a irregular xylem 1 that is impaired in the cellulose synthase 8 required for the synthesis of secondary cell wall cellulose. And this is the mutant in the beta subunit of the AGV1 protein that is a very susceptible mutant, also impaired in the cell wall. And this is our susceptible control because these are highly susceptible plants. Interestingly, the Arabidopsis response regulator mutant, as you can see here, show an enhanced susceptibility to the vascular bacterium Restaunia solanacearum, and this was a surprise for us. And you can compare here the C rating will mean a very high infected plant, and you can see here the blue line that is the allele compared to the wild type plant that is the gray line. As shown before with Plectosphorella, the overexpression line in this case is more resistant to Restonian than the wild type plant, and the overexpression of a line in the mutant background, as you can see here, almost restore the susceptibility, the enhanced susceptibility of the mutant to wild type level. As previously described by the group of Chris Argueso, in the case of Hyaloperonospora arabidopsidis, we don't see any significant phenotype in the single mutant in the AR6 gene, whereas the plant overexpressing the AR6 gene show an enhanced susceptibility compared to wild type plant. And these are again two controls of the experiment. So clearly this data indicate that impairing the ERR6 gene results in alteration of the resistant phenotype to two different pathogens. And it was surprising for us because based on the previous data of Chris and other groups, impairing of a single AR has no result in the alteration of the disease-resistant response. And this is a good indication that this particular member of the family should have a very important function in immunity. So the next that we did was just to perform a comparative transcriptomic analysis using Agilent arrays, and we identify around 205 genes in non-inoculated mutants plants that were differentially expressed compared to the wild type plant. And interestingly, a significant number of these genes belongs to gene ontology families that are related with defensive response, like response to chitin and other response to other stimulus, indicating that in this mutant there is in some way some constitutive activation or some immune response in comparison to wild type plant. So we check by QRT-PCR the expression of some of these genes that are upregulated in the mutant compared to the wild type plant, and you can see the result of this validation of the transcriptomic analysis in the lower part of this figure. And among the genes that we checked were some encoding transcriptional factor from the ethylene response factor family, like ERF6 and ERF1, and also some genes that have been involved and are really well described as genes activated by immune response after treatment with some MAMs like Phi1 or CYP81F2. So this is a good indication that in this mutant there is a transcriptional activation or some immune response. And we also check actually this is figure number 2 in the paper. So we also check the response of the mutant after infection with Plectosphorella cucumerina 1 day post-inoculation, and compare this with the response in infected plant. And what we found there is that there is a significant change in the expression of genes in the mutant compared to the wild type plant. Interestingly, there is a very high overlapping of the defensive response after infection with this pathogen. But as you can see here in this main diagram, there is also a specific response of the defensive response in the mutant compared to the wild type plant. Interestingly, among these, the genes that are upregulated at the infection with Plasmodium are some of the genes that you can see here in the left part. Some of the genes that are constitutively expressed in the Arabidopsis response regulator mutant, and these genes are also, as you can see here with the color, are also, some of them are also induced after infection with Ralstonia solanacearum, indicating that some of the genes that are constitutively expressed in the mutant also are upregulated upon infection with the two pathogens that we have seen alteration in the level of resistance in the mutants compared to the wild type plant. Interestingly, and you can see here from the genes that are specifically activated in the Arabidopsis response regulator 6.3 mutant allele upon infection with Plasmodium cucumerina, they are a gene related with a response to stress, response to abiotic stimulus, response to different compound like carrikine, hydrogen peroxide, and so on. So clearly there is a set of genes that are specifically induced in the mutant after infection with Plasmodium cucumerina. So we check if after this infection with the fungus, the expression of defensive genes will be altered in the mutant compared to the wild type line. I mean, in particular, marker genes from different canonical pathway, like the canonical pathway regulated by salicylic acid, ethylene, jasmonic acid, abscisic acid. Also markers for PTI or genes encoding protein related with the synthesis of secondary metabolites that we know that are very, very important for Arabidopsis resistant to necrotrophic pathogen, in particular to Plasmodium cucumerina. And as you can see here, when we compare by QRT-PCR analysis, the expression of this marker gene in wild type plant called 0 or in the mutant after infection with the fungus, we don't see a defective response of this marker gene in the mutant comparing to the wild type plant. Actually, the majority of this gene show a slightly enhanced expression in the mutant compared to the wild type plant, that is in line with this result that I have shown here, indicating that some genes related with immune response are slightly show a slightly enhanced expression in the non-infected plants compared to the non-infected mutant compared to the wild type plant. So interestingly, we also check what is the response of the mutant of the transgenic plant to treatment with different MAMs like flagellin and also chitin. And I will not go into much detail, and I will focus on the result shown here, just to show you that interestingly in plants that are treated with flagellin, the expression of some marker gene of the flagellin PTI response, like Fig1, is enhanced in the mutant, whereas this expression is in the transgenic plant is like a wild type plant. So in some way, some of the immune response activated by MAMs are slightly, the activation of the response are slightly higher in the mutant compared to the wild type plant. And in the transgenic plant, as you can see here in this analysis of the phosphorylation of MAP kinase, the response to different MAMs seems to be lower than the response observed in the wild type plant. So this may explain this phenotype that of the overexpressor line that show enhanced susceptibility to Plasmodium and enhanced susceptibility also to yellow peronospora, as was published by the group of Chris Argueso a few years ago. So how about cytokinins? Because this is a protein that is involved in the activation of the immune, in the activation of the cytokinin pathway. Well, to our surprise, we compare the expression, the constitutive expression of differential express gene in non-inoculated plants of the ER63 allele, and we compare with the published data of mutants impaired in the cytokinin pathway, like the double AHK1, HK3, and or the single HK1, and basically we didn't find any significant overlapping. And more interestingly, we also check the overlapping of the genes differential expressed in the R63 mutant or in non-inoculated plant or after inoculation with pathogen, and we compare this gene with the collection of genes that have been defined as the core collection of genes that are related with cytokinin pathway, that are these 75 genes. And as you can see here, the overlapping is very, very low or close to zero. And this is a very clear indication that the response that are regulated in the R63 mutant are not related with the cytokinin pathway, and this was a surprise for us. Actually, we also checked the level of different hormones in the mutant compared to the wild type plant. I will not go into much detail. And we performed a global metabolomic analysis. This was done by the company Metabolomic Incorporation, that is located in Research Triangle Park. And basically, with the exception of camalexin, that we know that are not important for the resistant to Plosphaerella, or gentisic acid, and some other minor metabolites, we didn't see any significant difference in the Arabidopsis response regulator mutant compared to the wild type plant, indicating that the constitutive response or the enhanced resistance to Plosphaerella is not related with the accumulation of other defensive phytohormones in these plants. One of the thing that we identified was that there were a group of genes related with cell wall synthesis and remodeling that were upregulated constitutively in the AR6 mutant. And based on also on previous data that have shown that the Arabidopsis response regulator could be related with or could be involved in the regulation of the protoxylem formation in the vascular system of Arabidopsis, we decide to explore if, like in other cases that we have in the lab, this mutation will affect the cell wall composition, and this will have an impact on Arabidopsis resistant to this pathogen, Plosphaerella and Rastreina solanacearum. And to do this, what we did was to extract a cell wall from the wild type and the mutant, and to perform different biochemical and cell wall analysis. And this analysis revealed that clearly the AR6 mutant show alteration in the cell wall compared to the wild type line. For example, here in the upper part, in the A panel, you can see this is the figure 3 of the paper, the comparative analysis by Fourier transmission infrared spectroscopy. So when in this graph, we are comparing the wild type and the mutant. So basically, the black line will be will correspond to the wild type values, and we are subtracting from this value the values of the cell wall from the mutant. And you can see here there are here some areas where the absorbance by this technology differs from the wild type plant. And this is related with lignin. This area of absorbance in F tier has been described to be related with lignin, homogalacturonine, that is one of the components of the pectins. Based on this, we analyzed the composition of the two fractions of pectins from this cell wall from the wild type and the mutant. And as you can see here, clearly, when we determine the monosaccharides in this pectin fraction from the wild type plant and the mutant, we found that there are significant difference in the composition of the polysaccharides from this fraction, because they are different, significant different in the amount of the different monosaccharides like arabinose, galactose, glucose, And as you can see here in the mutant, there is an enhanced accumulation of galacturonic acid that is the main component of homogalacturonan. And we can see here this in the pectin 1 and pectin 2 fraction. So we also determine the other fraction that I mentioned before from that is a main component of the cell wall, that is Emi Cellulose. And as you can see here, there are no major difference in the composition of Emi Cellulose between wild type plant and the mutant. And also we analyze the composition of cellulose and lignin, and as you can see here, there are no significant difference between the composition of the content of lignin and cellulose in these two mutants. So clearly the pectin fraction from the mutant differs from that of the wild type fraction. And interestingly, there is an enhanced accumulation of galacturonic acid that is the main component of homogalacturonan, and homogalacturonan is the source of one of the few glycan dams from plant cell wall that have been characterized in detail, mainly by the group of Giulia Di Lorenzo, that are the oligogalacturonides or OGs. So one interesting hypothesis could be that the RR63 mutant has a higher level of OGs in their cell wall. Just to check this, what we did was with this fraction that we extracted, we test the capacity of the cell wall fraction from the wild type plant and the mutant to trigger immune response. And we perform this type of analysis regularly in our lab. And for this, we use we measure different early immune response. And one of the immune response that we measure is the increase in cytoplasmic calcium. And for this, we use these transgenic plants, Arabis transgenic plant that overexpress the apoequarin in the cytoplasm of the plant cell. And this apoequarin, as you can see here, we just add the apoenzyme, the cholinteracin, and we can just have the aquirin. And if there is an increase in the calcium in the cytoplasm of this plant, after, for example, treatment with a MAM or after treatment with this cell wall fraction, this aquirin protein will bind the calcium, will release the cholinteracin, and this will result in the production of light that we can measure in a luminometer. And this is a very fancy technology that we use to determine the level of calcium after treatment with different compounds and also after treatment with different fraction, cell wall fraction, like the fraction that you can see here. And as you can see here in this panel, the pectin fraction from the cold cell wall clearly activate an immune response because we see this peak of calcium. But interestingly, the peak of calcium, or the calcium burst that we'll see when we treat these apoequarin lines with the fraction coming from the mutant line is clearly higher in the case of pectin 1 fraction than those that we saw after treatment with the same fraction from the wild type plant, indicating that in some way the pectin 1 fraction from the mutant may contain more DAMs or additional DAMs at the cold zero fraction. And to further demonstrate that this fraction contains or are able to activate the immune response, what we did was to check the phosphorylation of MAP kinase in this phosphorylation kinase assay, of the fraction coming from the pectin from the cell wall from Columbia at different time point, 10 minutes, 20, and 30 minutes after treatment with this fraction, and from the corresponding fraction, pectin 1, that activated strongly the calcium burst. From the mutant, as you can see here, the phosphorylation was enhanced after treatment with the mutant fraction compared to the wild type line, whereas in the case of pectin 2, we don't see too much difference. These are in the right part, you have the control with flagellins and with the OGs, or the degree of polymerization between 10 and 15 that also activate the phosphorylation of same kinase, MAP kinase, that the cell wall fraction, the pectin cell wall fraction from the mutant. We also check the expression of some marker genes, and as you can see here, in the pectin 1 fraction from the mutant is able to trigger the upregulation of the expression of the FIG1 and WORKI33, and this activation and also from the CYP81F2. And this activation was enhanced compared with that of the wild type fraction, and in some cases not quite high. And we performed the same type of analysis. Actually, with the pectin-2 fraction. Actually, the activation of the upregulation of some of the genes is at the same level that we observe after treatment with OGs or flagellin, as you can see here in this control panel that is in the right side of the slide. So in summary, the characterization of the Arabidopsis response regulator 6 mutant allow us to identify a novel mechanism of immunity that basically our hypothesis is as a consequence of the mutation in the Arabidopsis response regulator. There is some change in the cell wall composition, mainly in the pectin fraction. And as a consequence of this alteration, there is potentially the release of some glycans dams, and these glycan dams will be perceived by some receptor, and this will activate the immune response, and this may be the explanation that in the mutant plants we see a constitutive expression of some defensive genes. And this constitutive expression of defensive genes and also the activation of the immune response upon infection with Plasmodium varicella will result in an enhanced resistance to pathogens, in particular to Plasmodium varicella. In the case of Ralstonia solanacearum, we still don't understand 100% the base of the resistance, but based on the mechanism of infection of Ralstonia, it may be possible that this change in the vascular system, in the cell wall of the vascular system of the Arabidopsis plant will impact on the colonization or the enhanced colonization of Ralstonia by Ralstonia of this Arabidopsis response regulator 16 mutants. And I guess this is a very good example of that, starting from the characterization of one gene that is involved in the regulation of the cytokinin pathway, we reach to the point to identify a cell wall fraction that show enhanced capacity to trigger immune response in plants. So this should be the end of the history, but recently we have published the follow-up of this article in Frontiers, and hope that you will allow me just in no more than 4 or 5 minutes to explain what is the exciting discovery that we have done in the follow-up of this pectin fraction. So this, as I said, this was published in Frontiers in Plant Science. The first author of this article is Hugo Mélida. Also, Laura Babastete is co-author of this article. And we started with a very difficult task that was to try to reduce the complexity of the pectin-1 fraction by using chromatography filtration. And we obtained different subfraction of this pectin fraction from the wild type plant and the mutant. And with this subfraction, basically the D subfraction are just the salts of the extraction. So just we ignore this. And with this subfraction, again, using the calcium sensor lines, we check the activity of this, the wall fraction or the different subfraction. And interestingly, we found that both in Columbia and Arabidopsis response regulator 6.3 mutant, the subfraction C was the most active of this subfraction. And what we did was, or what we decided was to characterize this subfraction C. This is a very difficult task because this is still a very complex fraction. And when we analyze this fraction, that is the most active one, we found that this fraction is enriched in glycans, that the main component will be arabinan or silanx, because this fraction were enriched in these two monosaccharides compared to the fraction from the wild type plant. And still we see an enhanced accumulation of galacturonic acid compared to the fraction of, in this case, will be the subfraction C of the wild type plant. And based on some MS analysis, we determine that in this fraction, there were oligonucleotides that are composed of pentose. And based on our knowledge of the cell wall, we thought that these oligonucleotides could be derived from 2 different types or 3 different types of polysaccharide. That will be arabinans, arabinosilans, or silans. And what we did was just to analyze the capacity of some of these arabinosilan, silan, or arabinans to trigger immune response in plants, compared with the chitin using, again, this calcium sensor system. And when we apply this compound, you can see here that only arabinosilans have some activity on this, on triggering the calcium burst. Since these are very complex structure, we digest the active structure, and after this digestion, we obtain different fraction. And interestingly, some of these fractions that you can see here, the fraction number 4 and 5, were highly active in triggering this calcium burst. And we determined by mass spectrometry the composition of this fraction. And you can see here, this fraction mainly contain oligosaccharides of a DP of 5. So DP of 5 means that we have 5 pentose that, and based on this finding, what we did was to check several oligonucleotides, several arabinosilans that are commercial available, and we check the activity of these oligonucleotides to activate or to trigger immune response. And you can see here we have 4 different structure. I will not go into much detail, and to tell you a very long history just in a short way, just to mention that this XA3XX was the most active compound from this pentose, oligopentose that we tested. And this compound was able to trigger the immune response, just as you can see here by the calcium burst compared to the other compound. And I will skip this. And just to be sure that the activity of this compound is not due to a small contaminant of peptides and so on, we treat the compound with proteinase K. And as you can see here, when you compare with flagyellin treated with proteinase K, in the case of flagyellin, we lost completely the calcium burst, as you can see here, compared with the flagyellin. Whereas in these compounds, there like chitin on this novel oligosaccharide that we have identified, when we treat with proteinase K, we don't see any reduction in the activity. Whereas if we treat this with, we hydrolyze the glycan, we see a strong reduction of the activity. So this is an indication that the activity of these compounds, this commercial compound, is clearly derived from this oligopentose. So as I said before, we have purified also this compound, and this compound is here. And in summary, what we have identified is this novel DAM that is derived from arabinosilans from the cell wall from plant, and that we guess that is the component that is explaining the enhanced activity of the pectin fraction of the arabidosis response regulator 6 mutant. So interestingly, the activity of this compound is independent of THER1 and BAC1, and you can see here in the calcium, measuring the calcium burst of this compound compared with flagyellin. So this compound is able to activate the calcium burst in apoechorin wild type plant, or apoechorin lines in the THER1 background, in the mutant background. And as you can see here, this compound is able to trigger the MAP kinase phosphorylation in wild type plant THER1 and BAC1 mutant. And we perform also a transcriptomic analysis, and in wild-type plant, as you can see here, when treatment of plant with this compound, there is a significant activation of immune response gene in the plant. And more interestingly, when we apply these compounds to tomato and pepper 2 days before infection with either the bacteria Pseudomonas syringae or the necrotrophic fungus Sclerotinia sclerotorium, what you can see here is that in the plants treated with this compound, the disease, the growth of the bacteria or the disease symptoms are clearly reduced compared to the wild-type plants. So, and this is a very interesting result, because this means that we can use this compound to trigger immune response in crops. So that means that crops are able to perceive this novel DAM, and also that this compound could be used as an alternative in crop protection to the use, for example, of fungicides. And this, I guess, is the conclusion of this long history with through the Arabidopsis response regulator 6. And this also is connected with the title of this press release that I mentioned at the beginning, that was done, that was coordinated by Ashley. And clearly it's a good indication that we can use the cell wall to trigger immune response, or cell wall components to trigger immune response, not only in Arabidopsis, also in crops. And this has a very high interest as a solution for sustainable agriculture. With this, I will just summarize my presentation, just the take-home message. The first one will be the cell wall integrity composition alteration, like that generated by impairing the Arabidopsis response regulator 6 gene expression, can modify the natural resistance of plant to microbial pathogen. And this is clearly shown by analysis the mutant and the transgenic plant. That impairment of the Arabidopsis response regulator 6 expression result in the constitutive expression of non-canonical deficient pathway, and also genes, and enhanced resistant to pathway and genes, and enhanced resistant to the necrotrophic fungus Plasmodium Molina. That the impairment of the AR6 gene expression result in cell wall alteration that may contribute to Arabidopsis resistant to the vascular bacterium Rastreina solanacearum. That the pectin fraction from this mutant trigger more intense immune response than those activated by similar wall fraction from wild-type plants. And this suggests that this fraction contains higher level of DAMs than wild-type plant or novel DAMs. And based on the analysis of this fraction, we have identified a novel cell wall-derived DAMs. There is a pentose-based oligosaccharide that can explain the observed enhanced PTI response and the disease resistance of the Arabidopsis response regulator 6.3 mutant. And it's interesting to conclude that the molecular basis of plant perception of novel glycans have to be determined, in particular of this, another that have been published recently in the literature. And this is a clearly understudied field, that plant cell wall is a key component of plant immune system and a source of uncharacterized DAMs, glycans, as we show in this with these two papers. And the cell wall derived DAMs, we guess that could be developed as a novel crop protection technology for sustainable agriculture. And actually, there are several companies that are very interested in this technology, and we are collaborating with some of them to explore the potential of using these novel DAMs and in general glycan-derived MAMs and DAMs for the activation of a natural crop immunity response. And with this, I will finish, and I will be delighted to answer your question. Just only to acknowledge again the wall team. We call this Paredianos, because the wall in Spanish is pared. And the Paredianos from my group are Laura Bacete, Hugo Mélida, Gemma López, Diego Rebaque, Piti Fernández Calvo, Eva Miedes, Irene del Hierro, and Lucía Jordá. And also to acknowledge the collaboration of our and the contribution of our external collaborators, Deborah Kofner and Nicolas Denans, Vincent Boulon, Dominique, Fabian, Michael Hahn, and Julia Santiago. And with this, I'm finishing, and I will be delighted, as I said, to answer any question that you may have.
Thank you, Antonio, for
Sorry, this was,
an
I guess this was
excellent
too
talk.
long, maybe, but it was 2 papers in one. So,
Well, thank you for an excellent talk. It was really interesting, and I think interesting to see where this all went, what the arc is, how you go from first discovering this phenomenon to breaking it down to what is the molecule to seeing what is the point. So it's a very, very nice to see that entire story arc, and I really appreciated that. So we have lots of questions.
okay.
If you have a question that you would like us to read, to respond to, please type them in the Q&A. You'll see that box down below. That's what we will be following. So please enter your questions there. And I will start with the first one, which is from Ralph Panstruga, who asks, can you comment on what more resistant or more susceptible means? Does that mean more symptoms or more biomass, or what? Something else?
So that's a good question, Ralph. So, and you know that this is one of my comment and regular comment in my reviews. Okay, so some of the paper, for example, that you sent to me. So I guess it's important just to quantify the biomass. So in our case, we always quantify biomass. In the case of Plectosphorella, we can quantify the fungal biomass by determining the amount of DNA of the fungus in the planta. In the case of Hyaloperonospora, we determine the conidiophores formation or the the production of spores. And in the case of Rastonia, these experiments were not done by us, were done by our French collaborator. They determine the wilting symptoms, but there is a tight correlation between the wilting symptom and the level of bacteria in this case.
Excellent. Olga, do you want to read the next one? Oops, I think
I'm still muted. So
yeah.
it's again from Ralph. So it's again asking how about the phenotypes of your wall mutants with erythrithe?
Well, this one, I guess no difference, but I don't recall now, Ralph. I can send to you an email with the answer, but I don't recall now. So I guess it's like wild type plant.
So we have another question here. Sophia, whoops, it just moved. Sophia Garnica, and she asks, could cell wall mutations be used to develop resistant varieties?
Yeah, so this is a exciting question. So actually, I will suggest you to visit our bioarchive preprint because this is one of the hypotheses that we are just discussing there. Because actually, what we did there was to find correlation between the level of resistance of the cell wall mutants and to different pathogens, and clearly we have seen in some, I mean, the level of resistance and the fitness parameter of these mutants, and we found a very surprising result. That is that in the case of resistance to vascular and necrotrophic pathogen, when you are testing cell wall mutant, you see or you find a negative correlation between the level of resistance and the fitness parameters of the plant. That means biomass and seed production. So clearly indicating that for Arabidopsis, to get enhanced resistance to necrotrophic and vascular pathogen has a fitness cost, at least in under our grow condition. But this is not the case for the biotrophic pathogen Hyaloperonospora parasitica, and Hyaloperonospora arabidopsidis. And this is interesting because in breeding programs, it's very easy, let's say, to find traits for enhanced resistance to biotroph, but this is not the case for a necrotroph and vascular pathogen. And this may be because our hypothesis, because maybe the breeder is not selecting this tiny small plant because they show an enhanced resistance, but they will not have an enhanced production of seeds or fruits. But based on this analysis, we have identified some cell wall mutant in which we don't see this fitness cost. And clearly this gene will be an interesting trait for breeding programs. Thanks for this fantastic question, because this is one of the hypotheses that we are working with.
So I move to the next question now. This is from Belal Hossain, and he's asking if your research is applicable in terms of plant viruses infection.
Well, this is another very interesting question that we are now checking. So this is a specific program that we have now supported by this excellent program that the center got. And we are checking the resistance of the majority of this mutant, this core collection of mutant, to viruses, to necrotrophic bacteria, to every pathogen that we have around. Because we want to integrate all this data and to develop models. Also in this bioarchive paper, they are very interesting modeling to try to find correlation between a specific change in the cell wall composition and the level of resistance to particular pathogen. This is a crazy idea, but just look at this paper. So, because they are very interesting correlations.
So Saeed Mubashir asks, is genetic resistance possible for necrotrophic pathogens which actively kill the host tissue and doesn't allow them to express their resistant genes?
Oof. Difficult question. So, so I guess, well, there are some examples now in the literature of mutants of some QTLs that compare a broad spectrum resistance, including to several necrotrophic pathogens. So, for example, there are very fancy publication of a group of receptor protein that is called, these are called this tandem protein kinase receptor. And some of these QTLs, because these actually some of them were identified in crops, confer broad spectrum resistance, including to several necrotrophic pathogens. We also have another example in our lab that is the Jodan MAF3 kinase that is part of the ERECTA pathway, and the constitutive activation of this Jodan MAF3 kinase confers broad spectrum resistance to several pathogens, including several necrotrophic pathogens. So I guess that we need to have, particularly the young PhD student, open mind. So because I guess that there are a lot of non-canonical pathway, immune pathway that still we need to discover, and we should be very open to find novel things. So actually our paper is an example that we just initially wants to characterize the contribution of cytokinin pathway in the resistance to several pathogens, and we realized that probably this is not fully related with the cytokinins. It's more related with the effect of cytokinins on the cell wall. I mean, it's an effect, but it's an indirect effect, not a classical, I will say cytokinin effect. And that's it.
Yeah, so I guess with this you answer also his next question, which was about which kind of necrotrophs have been used. So I think you comment on this, but link on this, I'm stepping in because I mean, you made a really nice correlation between your response regulator ARR6 mutant with pectin, right? But I mean, this mutation leads to many different changes. So did you test pectin mutants? So like Quasimodo, or I don't know, I'm not too familiar with that, but probably there are some in Arabidopsis. So would you see then, or would you expect, or did you see the same phenotype? I mean, are you really sure that this is?
Well, so there are two angles in your question, Alga. So the first one is we have tested other pectin mutants. Well, we have tested a few of them, okay? And I guess that there is in general, there is an oversimplification of the one mutation that impact pectin on the structure of the pectin fraction. So based on our experience, yes, a small as a mutation that in principle will change, yes, or will affect a small structure of the pectin fraction has a significant effect in other component of the pectin fraction of the cell wall. So we have tested several pectin mutant, and we see a lot of different different phenotype. Actually, in this list of mutants that I mentioned, that is in the bioRxiv article, they are pectin mutants. But we have not focused too much on this because the group of Giulia Di Lorenzo was already working on this. So, and we don't want to compete in this in this field. But at the end, we reached one point in which the Arabidopsis response regulator 6 was affected in pectin, and we thought that this differential activity will be related with more OGs. But we can tell you that this is not the case because I have no mention. But the OGs pathway require the one particular group of MAP kinase that are the ANP, and we tested this ANP, the cell wall fraction of the Arabidopsis response regulator, and the activity is similar to the wild type plant. So we don't see that the activity is impaired in this mutant. So basically, we don't see that OGs are the dams that are enriched in the Arabidopsis response regulator that are probably this other compound. That could be the only one, or who knows? We are characterizing in more detail this fraction, but this is a difficult task because these are very complex mixture of a structure.
So I'm going to suggest, since we've already answered 2 questions by Ralph Panstruger, it's another good one, but I think we need to move on to somebody else. Claudia Martinez Anaya asks, how could the mutation of ARR6 gene affect the cell wall composition? And so maybe you could just recap that a little bit.
Okay, so there is a very nice paper from the group of Thorsten Hellman describing this alteration in the synthesis of activity of cytokinins clearly impact on cell wall integrity. And also it's really well known that cytokinins are regulating some developmental process that involve the synthesis of the cell wall. So clearly we impair the Arabidopsis response regulator 6. This will have an impact on the synthesis of the cell wall, and this is the result of this will be an alteration in the cell wall. So I mean there are already several data that indicate this, and also there is some data published in our paper that mutation in the Arabidopsis response regulator 6 result in the constitutive expression of cell wall ready genes, and these cell wall ready genes that are constitutively expressed clearly will impact on the composition of the cell wall.
So the next question is from Gumi Lang, and the question is, do you think that plants have different pattern based on cell wall remodeling to recognize pathogen infection?
Could you say again, Alga, because I didn't
Yes. So do you think plants have different pattern based on cell wall remodeling to recognize pathogen infection? Not sure how to interpret this question, but I guess.
Well, I guess that if I understood correctly the question, this is a key point. So the question is how this change or this collection of glycan dams derived from the plant cell wall are perceived. Okay, and this is actually the main topic of my group now. This is a very interesting question because actually from the 700 potential pattern recognition receptor in Arabidopsis genomes, almost half of them are related, half of them have ectodomains that are not in principle related with the perception of peptides, because these are ectodomains related with as I mentioned in one of my slides, that have a similarity with lectin domains or other domains that in human immunity have been described to perceive glycans. So this is another interesting discussion. What is the knowledge of the perception of glycans by the innate immune system in animal and the innate immune system in plant? That this is something I'm missing because in plants, the number of receptors that have been well characterized are just 2 or 3, whereas in humans, we have 2 dozens or more. And this is missing because the cell wall of the plant is full of glycans, and we still don't know how all this alteration of the release of this potential dam are perceived. And we guess that this is really an explored field, and actually I guess I will just stimulate to work on this field. If you want to do in my lab, you will be really welcome, because this is one of the areas of our interest right now, because this is clearly understudied.
So our next question from Dr. Asif Mehmood is a very general one. What is the role of phytohormones in plant immunity? And I think that might be too big for the Q&A. But you did have a beautiful figure on that earlier that was from your earlier review, I think.
Well, I don't know if this is in my earlier review or it's just part of the thesis of Laura, but since you will have the presentation, or we can send to you, or you can look for this fancy figure we can send the figure to you, and you can just spread the figure.
And this is actually a good point, is that I know people are enjoying interacting with the authors at the end of the presentations, but you know, the corresponding author's email is there, right, as part of the affiliations, et cetera, and it's there so that you can email them with questions, right, and observations. So you should definitely realize that a paper is not a static thing, but that it is something you can still be involved in.
Yep.
Okay.
I will be very happy to answer this. So you can do just one by one better, okay? So.
So there is a next question from Panstruga, but I think it's an interesting one, so I'm going to ask it. So what could be an upstream of AR6? So he's commenting that at least in bacteria, response regulators act together with histidine kinases and form a signaling module. Is this also true in plants? And so what is, in your opinion, upstream of the AR?
So we don't know, because this is a very interesting discussion that I had with Chris Argueso in the last MPMI meeting in Glasgow, and how to integrate this in the immune response. And the answer to this is I don't know. So the only connection that we see is that there is one of the cytokinin receptor that also has been described as a cell wall integrity sensor. But at the same time, when we compare the gene expression of mutants in this cell wall sensor and the arabidopsis response regulator, we don't see overlap. So, but also we have to keep in mind that arabidopsis response regulator is a big family with a lot of overlapping. I guess we were very lucky to see a phenotype with the AR6 because we use the right pathogen. And this is also a very interesting point to address here, is that if you just focus on one pathogen, sometimes you will not see an effect, phenotype in your mutant. So in our case, we always check our mutants with 3-4 pathogens that we have in-house. And this is very important because if you just analyze different pathogens, the chance that you will see a phenotype clearly is higher. And also this will contribute to characterize the molecular basis of the resistance mediated by the gene impairing this mutant.
Yeah, and just to follow up on that, my own thinking is you have this histidine, relay, phospho-relay, and it ends up on one class of the RRR genes, which then transcriptionally activate the class of response regulators that includes ARR6. So I guess one question is, and since there's so little overlap between what it regulates and cytokinin, do you think that there's another input that regulates the AR6 transcription that's distinct from cytokinin signaling?
Well, there is a very interesting result in one of the supplementary figures that I didn't mention, but in the overexpressor line of the 35S AR6, when we treat with the flagellin, there is a downregulation of the expression of the AR6 expression, and this is something that was astonished So fortunately, the reviewer were not asking too much about this because we cannot explain, but this is reproducible. So for some reason, even in 35S lines, the level of expression of the ARR6 transcript is reduced after flagellin treatment. And this is a very exciting result because this is indicating in line with your comment, Jane, that there is an additional level of regulation that we don't understand. That's my impression. But
okay, our next question by Olalua Ajayi has been already answered. So let's move on to Eugenio Sanz Garcia. Do you find any other phenotypic difference between Columbia and the ARR6 mutant?
No, at the developmental level, they are no different. Actually, the overexpressor line are like wild type plants. For example, this coming to some of the previous questions regarding the potential use of the cell wall for a breeding program. So we initially thought that the Arabidopsis response regulator will be a very interesting target for breeding because there is no any effect on fitness. But initially we found that the plants were more resistant to Processorella. Oh, there's a good trait, but good seed production, good biomass. But later on, we received the data from our collaborator that performed the assay with Restonia solonacearum, and we found that we're more susceptible. So we were performing some selection in tomato and with a company, and we stopped to do this because clearly this will be not a good idea. But basically in this case, there are no difference in the fitness and other developmental phenotype difference compared to wild type plants.
So we have now a question from Garrett Noon. First, the comment, cool work.
Thank you.
And then the question, have you examined the susceptibility of ARR6 mutants to Pseudomonas syringae?
Yeah, I guess this has been done by the group of Chris Argueso, and no difference, or a small difference, slightly difference, as far as I recall. Okay.
Prashant Pawar, thanks you for a nice presentation. And then also asks, what is a probable receptor for XA3XX?
Well,
you can tell your next Nature paper.
No. So we don't know yet. So we know that it's not third one independent. That this is so we are working on this. So we are selecting. Well, we are using different approach to try to determine this, including some in silico approach. So, but this is part of another paper that we are working in. So, but it will be tough to identify this type of the receptor for this compound. But we are working on this.
Maybe next year.
Next time.
So from Michael Hanna, you reported an increase in the GAL-A component of the mutant cell walls, yet GAL-A containing molecules do not show up in your fractionation. So care to comment?
Yeah.
Where is it?
Hi, Michael. Nice to see that you are there. So this is one of our collaborators. So, and you see, he's just asking tricky questions. So, you are right. And in the fractioning, we don't see this increase in GAL-A, and I mean, compared to the pectin fraction, and we guess that probably is because we lost some of the polysaccharides in this fractioning, or we don't know the explanation, or maybe there is a lot of GAL-A small OGs or oligogalacturonide saccharides that are in the D fraction, but this D fraction we were unable to determine because there are a lot of salts. So we don't know. At least we see that there is an enhanced accumulation of GAL-A in the mutant compared to the wild type. But you are right that compared to the pectin-1 crude fraction, the level of GAL is lower than in this fraction. So we don't know.
Sorry, the next question is, how do I get the paper? How do I get the presentation? And Ashley has pasted those links into the chat, so you can go there and find them. But you could also go look at, I think it was the May issue of MPMI. That's where this paper was. A lot of other good papers there. And also where you went to register for this is also where you go to find the recording, and that should be up in a couple hours. We're pretty fast on that.
Mm-hmm.
So, and the next question is from Yegret Kaur. Does treatment with novel damp trigger defense response in the absence of the pathogen?
Yeah, the answer is yes. So you just look at this Frontiers paper, you will see that you can see response in the absence, and also we have determined the response after infection, and also you can see that there is a response before and after. So actually the result with the, maybe I didn't mention, the result with the enhanced resistance in crops treated with the arabinoxylans, we treat the plant 2 days before inoculation with the pathogen. So, and in this particular article, we check the expression of some tomato marker genes, and these tomato marker genes are induced after treatment with these arabinoxylans before infection with the pathogen.
Okay, so the next question is again from Michael Hahn.
Okay, Michael, thanks.
It is a detailed question here. What is the molar response of the arabinoxylan in comparison with other oligosaccharide inducers, such as the oligogalacturonides?
Okay, this is a good point. I didn't mention the concentration, but they are very similar. So this is in one of the slides that I showed it. We always include the OGs as a controls, and the response are more or less similar. So it's in the same range that OGs. So I will say that these are active compounds, but we have now in the lab some glycans that are clearly more active than these compounds. So and I guess that still we will be able to identify some of these oligo structure with some decoration, acetylation, methylation, some decoration that will be more active than the structure that we are now working with. So, but the activity is very similar to OGs.
Okay, then there is a nice comment, best presentation and nicely done from Marena Farok, but no question. So she was just happy. And there is a question that I'm not sure I can interpret in the right way, but this is from Prasant Pawar. Are there any changes in plant cell wall of the XA3XX elicitor response?
That's a good question. I guess this is also one of our collaborators, I guess. We have not tested this, and this is a very interesting point. So this is something that we are now planning to test. So just to determine the cell wall alteration after treatment. The problem with this is that when you just treat with one of these compounds, and it's very important, and actually this is included in the discussion of the MPMI paper. So it's important for some of these compounds to have to have this compound formulated, like in the fungicides. So because some of these compounds, if you just apply is dissolved in water, in some cases they you don't see any activity. So because this compound has to reach the surface, had to be there. In some case, some epiphytic microorganism can use this as for food, and this is a very interesting balance. So the reason we have not done this is because this is no easy task with at the biochemical level. So we guess that using the function collection of the Michael Hahn contribute to generate and the collection of antibodies that recognize the epitope from the cell wall from the CCRC. Maybe with this technology we can determine this change in the cell wall. But at the biochemical level, we have been trying to determine the change in the cell wall after infection with some pathogens, and the amount of tissue that you get is so tiny that to perform some biochemical characterization of the change in the cell wall after infection is no easy task. And we guess that after treatment with the elicitor will not be an easy task unless that we are developing novel technologies, or we are using this collection of specific cell wall antibodies.
Okay, our next question is from Juan Carlos Diaz-Ricci. Nice talk, Antonio. Since you have observed a calcium burst in the RR6 mutant when treated with FLAG22 or OGs, I wonder whether this process is accompanied by an oxidative burst.
No, in this case, the oxidative burst is very weak. So in general, with the majority of the glycans that we have identified in our lab, so right now we have a collection of, I will say, another 10 glycan structures that are active, and the majority of them we don't see ROS production. So I know that it has been described that after treatment with OGs, you see a ROS production, and actually you can see it, but it's very weak. But compared with other structure, we don't see ROS, with the exception of a couple of structures that we see a clear ROS production.
So, Antonio, actually, this is quite interesting, right?
Yeah.
Because normally calcium and ROS are coupled. Do you think is a peculiar feature of Arabidopsis? And if you test it on barley, for instance, so a monocot or another more responsive plant, because Arabidopsis is not particularly responsive, right? To this.
Yeah, I know.
Would
Well, I guess that probably you will have the answer better than me, because you have some of these tools in your lab. So no, we have been collaborating with a company that has the ROS production system established in several crops. And in general, when you compare, when we treat with some of these compounds that we know, for example, that are active in tomato and pepper, and you compare with the ROS and other immune response with flagellin, the ROS production in general is very low. So, and you are right that there is this general idea that calcium burst and ROS production are coupled. And in our case, we are not sure about this. So, I mean, at the end, if you just treat with, I will say, non-biological concentration, you will see a ROS production. But the question is, this is real or not? So, and I guess this is a very interesting question to resolve. So we are working on this, actually. So, but we can send to you some of this our structure, and you can test in your system, Alga. No?
Happy to test. So there is now a question from Caterina Rivera, and she's asking, have you seen the effect of the overexpression of ARR6 in Nudgy plants?
No. Okay. No, we have not checked this plant that I know that have been generated by I guess Chris Argueso group, but I'm not sure. But no, we have not checked this. And also,
I mean, there is this crosstalk with salicylic acid, and so it would be actually is a quite nice.
Yeah, no, I guess this is a very interesting point. But since we didn't, I mean, based on our analysis, in non-inoculated mutant or inoculated mutant, we don't see an effect on the SA pathway. So we don't see an overrepresentation of a lower upregulation of salicylic acid related genes. So we doubt that the effect that we see is related with the crosstalk between salicylic acid and cytokinins. I mean, we guess that that's not the answer. So this is not the reason.
Okay, the next question is somewhat general. What is the role of cytokinin in plant immunity?
Well, that's a good point. So before this paper, I will say that the data published was the data from Chris Argueso lab, and this was in my presentation indicating that cytokinin could interact with salicylic acid and in some way trigger immune response. And Arabidopsis response regulator will be 6 will be a negative regulator of this activity for the reason, when you overexpress Arabiosis response regulator 6, you have an enhanced susceptibility to a pathogen like yellow peronospore Arabidopsidids that requires salicylic acid for a pathway for plant resistant, Arabiosis plant resistant to this oomycete.
Okay, the next question is from Roger Innes. So we now know of many dumps and mumps that can be sprayed on crops to induce resistance. Are any of these being commercialized?
Okay. Hi, Roger. Very good point. Yes, there are several commercialized and some are coming. And in some case, my impression is that we have been using this technology for a long time, but we were not aware of this. Just to give you an example, there are a lot of products in the market that are algae extract, okay? And these algae extract are enriched in beta-glucans, as Alga knows. and some of these beta-glucans trigger immune response. So, and these products are working very well in agriculture, particularly in what we call specialized crop, like vegetables, and under very controlled conditions, these products are working very well. Regarding the synergistic effect, this is a good question, and we are checking now this. But this is a very interesting question, since chitin and X, the arabinoxylan that we have identified, are not going through the same pathway. At least the activity of the arabinoxylan is third one independent. If we combine, if we see a synergistic effect. We don't know yet, because actually there is a PhD student that will start tomorrow, and she will work on this. So thanks for the comment.
Okay, our next one, and I suggest we only do a couple more questions as we're getting towards more than an hour. So Grisha Hao says it's nice work and asks whether XA3XX can trigger a defense response in monocots such as wheat and rice.
We don't know. So this is will be very interesting to test. So we find some collaborator, we can test this. So,
and following up on that, I see a few questions further down that are asking if this is commercially available.
No, not yet. One of the reasons, I mean, we guess that this compound is very interesting, but based on our analysis, not the best one to commercialize. So maybe somebody will kill me for this comment, but and the other reason is this compound is no will be very difficult to commercialize. Is that I just described very fast the purification of this compound, but we have to produce this compound and to purify, and after that to apply into agriculture. We have to be very efficient to have a very cheap price for the compound to include in a bottle as a new product. I guess that there are other compounds like beta-glucans, as I mentioned before, that are easy to prepare, and that the activity in some cases is even higher than this one. So, and the other reason, I guess, maybe this compound will be not commercialized in the future is because this compound, we published this paper and we don't patent this compound because there are some previous patents published, and it will be very difficult to protect this compound. Well, now impossible because we published this to protect the compound, and this clearly can reduce the interest of some companies, at least the big one.
So I can move to the next question. Actually, is from my lab, from Balakumaran Chandrasekhar. So he's saying that there was a very nice presentation, and then he's asking, he's curious to know how xylan-based oligosaccharides, so pentose, got enriched in the pectin-1 fraction of the ARR6 mutant. Does xylan and pectin form any kind of network, thereby leading to the enrichment in the fraction?
Yeah, that's a good point. So this is actually one of the questions that we got from one of the reviewer of the article.
Not us.
So well, I guess that there are some data indicating that with some purification process in the pectin fraction they are silent, and we know this. Again, you just go to the bioarchive paper. There is a analysis, I guess, of 7 mutants with the collection of antibodies of the CRC, the Michael Han, and you can see there that in the pectin fraction, when you use antibodies just to for detection of silence, you see signal. So this is a good indication that this standard protocol that we use to separate the pectin 1, pectin 2, Emi Cellulose, and 1 and 2. So this pectin 1 is enriched in pectin, but there is other things there, and this is the reason we identify this compound. And the same in the Emi Cellulose, you see that still there are some rest of pectin there. So I guess that this is a general classification in the cell wall field, but it's not very accurate, because I will call fraction CDTA or more related with KOH, more related with the compound or buffer that we use to extract, and not because this generates this confusion. Like that's the reason in the second paper of the Frontiers, we are talking about CDTA fraction. We are not talking about pectin fraction, because I guess this is a mistake. So the people, since there are not too many people from the cell wall in this audience, I can say this. No.
So I see that there is one comment farther down that says, is APS and MPMI going to have more of these talks? And the answer is yes.
Yeah.
In 2 weeks, we're going to have a seminar by Jared Leboldis talking about his paper, Population Genomic Analyses Reveal Connectivity via Human-Mediated Transport Across Populous Plantations in North America and an Undescribed Subpopulation of Spirulina musciva. So something a little more population genetics and looking at the interaction of humans and the whole plant microbe interaction. And Ashley has just posted in the chat where you can register, same place you registered for this talk. And Alga, do you want to ask one last question, or shall we stop there? Stop there. Okay, I'm sorry we didn't get to answer everybody's questions today. There were some terrific questions, and you also have some suggestions in there, Antonio, for collaborations. So you should read through all the chat. And lots of very nice comments of people who really enjoyed the talk and the figures, and are looking forward to hearing more about the receptors. And I should say, for those who we did not have a chance to answer, I hope you will consider emailing Antonio Molina, and you can get his email off the paper, and ask him some of these questions. We've got some terrific questions here, and it's always so much fun to see how people are different people look at the same paper and see different things jump out at them. And I find that very, very interesting. So I'd like to thank Antonio for a wonderful talk and for staying here, for staying for a long time for questions, terrific questions. And thank you to Alga for being the co-host. And I hope you will join us in 2 weeks.
Okay, thank you very much to you and Alga, and also to Ashley, and thanks for this editor pick. So because clearly this has contributed to put the cell wall in the real world, the relevance that we guess that we'll have in the next year. And as I mentioned to you before, thanks a lot for this very great initiative of the virtual seminar and this editor pick. And I guess it's a great opportunity for a young student, and I saw also for senior students like Roger, Ralph, and Michael, and others. So, thanks a lot for the invitation, and it was a pleasure to contribute to this virtual seminar.